Hello, General Biology, and welcome to the second in the screencast series for Chapter 7, Sections 7.3 and 7.4, and of course, including the cell wall and the cell membrane. And we're going to talk more about the cell membrane as I had mentioned. So in the last screencast, I gave you the term phospholipid. And I want to talk about that in a little bit more detail, and I might flip it back and forth to the picture above as we're talking about this. So a phospholipid is made up of a phosphate group, which we know are phosphorus and oxygen, and lipids, which we know are fats. It had, as I described, a polar head, and that polar head is made up of the phosphate groups. And what we know about polarity is having a positive side and a slightly negative side. What they do is they attract. So the phosphate groups are groups that attract water. They're polar, just like water is. And we call them hydrophilic. Hydro means water, and philic, if you know having a philia, Philadelphia, the city of brotherly love. Philia means love. So the heads are water loving, and they just pull water in. They just want to bring that water in. The tails, on the other hand, are non-polar tails, and they're made up of the lipids, which makes sense because lipids are not soluble in water. They don't like water. They repel water. So we call these non-polar tails hydrophobic. So again, hydro is water, phobic, phobia. What do you have when you have a phobia? Right, a fear. So these are water fearing. So those non-polar tails, they want to force the water out. So when we come back and we look at this membrane and we see right here that these polar heads, these hydrophilic heads, they're on the outside of the cell and on the inside of the cell, they're going to be pulling water in from both sides so that the water is constantly flowing back and forth. And these non-polar hydrophobic tails that we see in here, they don't like the water, so they're swooshing the water out so that we have a constant, even flow of water into and out of the cell so that the cell can remain stable. And what we're going to learn at the end of the chapter in homeostasis, which means balanced. It's going to be in equilibrium and balanced. So that's why we have these phospholipids. The polar heads are going to be pulling the water in. The non-polar tails are going to be pushing the water out. So as I mentioned, the heads are on the outside of the membrane. They attract the water. The heads pull the water into the cell. The tails are on the inside, and they're going to repel the water. And they're also going to form this oily layer inside of the membrane to keep water out. Now, that seems to be counterproductive, don't you think? But why do we put lotion on our hands that has oil in it? To hold the moisture in, exactly. And that's what we're trying to do. We don't want the cells to dehydrate. We want to keep that moisture in. So that oily layer actually protects in both ways, where it doesn't let too much water in for the cell to explode, and yet it keeps enough moisture in that it keeps it hydrated. Now we talked about the membrane proteins, and those were these little kind of brownish structures that you see here. And there's different kinds of membrane proteins. Structural proteins, which I mentioned as the keystone, where they're going to hold the membrane together, and transport proteins that are going to be used to move materials into and out of the cell. And some of these proteins are going to require energy to move things into the other cell. And others are just going to be kind of like an opening in a fence where they're going to allow materials just to move into the cell with the flow of the water that's being pulled in. When we use a protein, using it is called facilitate. So we're going to facilitate. We're going to use something. It's going to facilitate the movement of materials into and out of the cell. And you're going to see that that's going to be important in just a few minutes. So we have different kinds of proteins. Some of these proteins act as channels, and some of these proteins are going to act as pumps. The channels are going to be like that opening in the gate where it's going to be able to come through, and pumps, just like it sounds like, it's going to pump. It's going to do a pumping motion to move the molecules into the cell. So let's talk about those carbohydrate chains that are attached to the proteins, because these are significant. And some of this information that I'm going to give you right now, not all of it is found in your book, so it's really important that you're listening to this. So some of these proteins have carbohydrate chains that are attached to them. They are called glycoproteins, and they really play several roles in cell interactions. One of the things that they're going to do is work with the immune system. The pattern that they're arranged in will recognize if other cells work together with them, or if they're invading cells and they need to protect them against the invading cells. So that's part of the immune system. Another way is to communicate between the cells. So if it's the same type of cell, it's going to have the same type of carbohydrate chain pattern. If it's not the same type of cell, it's not going to have the same pattern. So if it lines up and it does have the same pattern, then they know they work together and they can communicate that. They also are going to be used to attach the skin cells together. We saw the chains and the fibers in that Harvard Inner Life of the Cell video as that white blood cell was moving through. So that's one of the things that it can do. Another thing that these glycoproteins can do is help stabilize the protein within the membrane. They can be used as a cellular addressing system. So, we have a certain address to identify where we live. Well, they have a certain pattern to identify what kind of cell type it is. And they also determine where the proteins are going to go to make sure that they're in the right location. So I have just another little picture or illustration of this that shows the glycoproteins. And this talks about why when people get cell transplants, or not cell transplants, but organ transplants, why it can be difficult for the body to accept the new organ that's made up of cells, because they're not recognizing the glycoprotein pattern. So they wind up giving them anti-rejection pills, or medicines, so that it doesn't reject and attack it with the white blood cells. So I thought that was interesting, and I threw that in there for a little bit of trivia. We also have carbohydrate chains that will attach to the lipids and will attach to the cholesterol, and they are referred to as glycolipids. And glycolipids, again, they have the carbohydrate chains. They're used for recognition and receptors for other chemicals, and at the end we'll understand what we mean by receptors for other chemicals as they go through. So based on these characteristics, and I'm just going to kind of go back here and look at this picture again. Based on the characteristics that we see here, that makes this membrane selectively permeable. And selectively permeable means that it's going to allow certain materials to be able to pass into the cell and out of the cell, and it's going to keep other materials from being able to go into and out of the cell. I kind of drew this, I made this picture. These are the polar heads. So if we were looking down on top with the polar heads, notice that, they're round and there's spaces that are formed in between. Because of that structure, it forms these spaces, and if the polar heads are pulling water in, and maybe we've got a larger glucose molecule, that glucose molecule is going to be able to fit through this space and make its way into the cell so that it can be used by the cell. So it creates these natural openings that you see here, and that's what makes it selectively permeable. Your screens, your window screens that I talked about, are selectively permeable. The spaghetti colander that we mentioned earlier is selectively permeable. It holds the spaghetti or the noodles in, it allows the water to go out, and that's what this phospholipid bilayer does. All right, so again, I think I'm going to end this a little bit early, 11 minutes in, because we have a video of the amoeba sisters that I want to start off with the next section. All right, see you in a little bit.